Good evening, everybody, and welcome to IF Oxford, the Science and Ideas Festival. My name is Cathy Rose. I'm the festival manager, and I will be hosting this event tonight with you. So tonight's event is behind the scenes at a primate lab. This is something that I've been really interested to find out about as a zoologist. The discussion tonight will be telling us how and why rhesus monkeys are being used to help answer some really important research questions, including how we optimize learning. We're going to see how the monkeys complete adapted computer-based tasks that are used to assess learning in healthy humans, but also those who have experienced stroke. And the event will highlight the latest findings in brain adaptation and learning after injury. Anna is an associate professor at the University of Oxford's Department of Experimental Psychology, and she's the principal investigator in the Thalamus, Cortex and Cognition Lab. Bit of a mouthful there. I'm sure she'll explain to you far better than I can what that means. So over to you, Anna.
Thanks, Cathy. Good evening, everybody, and thank you very much for joining us. As Cathy said, I'm an associate professor in experimental psychology here in Oxford University, and I'm the PI with Wellcome Trust funding for my research lab. And we look at how the thalamus and the cortex are interacting together, but also other areas as well, other subcortical areas, and we're interested in that and cognitive processing, things like learning, memory, and decision making. I'm here tonight with some of the people from my research lab. So one of the people here with me is Stuart Mason. He's the main primate trainer here. Well, one of the main primate trainers in the primate unit here in Oxford.
Hi, well, I'm Stuart Mason, and being one of the main primate trainers here at Oxford, I really enjoy working and training the monkeys using positive reinforcement techniques here.
And then another person who's joining us tonight is Juan Carlos Menendez. Juan Carlos, would you like to say hi? You'll just have to take your mute off. Not sure how.
I think that's unmuted now. Hi, everyone. Yeah, so I'm a postdoc working with Anna. I'm originally from Mexico, where I did my PhD. And I've been doing studies in neuroscience using different techniques. And currently I'm working with neuroimaging in the primates. So that's what I'm going to be talking about.
And the other postdoc who's joining, thanks Juan Carlos, the other postdoc who's joining is Brooke Perry.
Hi everyone, my name is Brooke. I'm also a postdoc with Anna. My research focuses on electrophysiology in the monkeys. Yeah, and I'll be sharing some of our results with you tonight and what we do.
All right, thanks Stuart, Juan Carlos and Brooke. Cathy, are we going to run the poll now? We have a poll that we would like to ask you. It's an anonymous poll. You won't be linked to it in any way in terms of your responses, but we'd be really interested to hear. You don't have to reply, of course, and it won't be shared in public with anyone, but it's just to be able to get an understanding, and then we'll ask you a few questions after the presentation as well. So there's 2 questions now, and there'll be 3 after the presentation. If you'd like to join in, it would be very much appreciated. So the way you do that is you just click on the multi-choice question. We can't obviously do it, so I can't demonstrate that to you, but you just click on the one. The first question is, how well informed do you feel about animal research in the UK? And there you make a decision. And then the second question is, how do you feel about animal research? And again, you just make a selection. And thank you again for participating.
So I'm just going to give another few seconds for people to finish answering. Okay, I think that is most people who've answered. So I'm going to end the polling in about 10 seconds.
Oh, thank you again, everybody who's participated. It's very much appreciated. Okay, so as Kathy's indicated, tonight we're going to be telling you a little bit more about how and why we study what our brain is doing while we're learning things. And we use non-human primates that we'll refer to as monkeys from now on, and humans in order to be able to do that. We're going to share with you some of our latest results that have just come out in terms of publication this week as well. But firstly, during our presentation, we just need to clarify a couple of things. We'll be sharing with you some videos, some data, and some animation. Please be assured that the videos and the data that we're actually sharing are, sorry, Kathy, what's happened with your hand? Be assured that those are only being filmed during the course of the normal procedures that we're carrying out with the monkeys for their experiments. And secondly, the experimental research that we do has to be regulated, and in the UK that's regulated by the Home Office. So all of us working with the monkeys need to have our own personal license in order to be able to authorize us to conduct the experimental procedures with the animals. We need to sit exams to be able to do that, and to hold a personal license, you need to carry on with your training. You need to keep up to date with the latest developments and refinements, replacement and reduction. In addition, our work is also authorised by a project licence that I'm responsible for, and again, the Home Office authorises that. It's a legally binding document that is signed off by the Secretary of State, and for me that was Helen Clark. No, it wasn't. It was Theresa May, I beg your pardon. To apply for a project licence, my work is subjected to rigorous and extensive reviews and assessment. Neuroscience research involves any animal, particularly with monkeys. There'll always be some kind of approval that is needing to ensure that there's no alternative to this to be available in terms of the animal species that is most appropriate to model these behaviours. The named veterinary surgeon, the named animal welfare officer at the institution must approve this before it can be then reviewed and approved by the local institute at the Animal Care and Ethical Review Committee in Oxford here and at other universities as well, depending on where you're based. Then the local Home Office inspector and a subcommittee of UK Home Office inspectors with primate experience will review it and pass on their assessments. They may ask you to change certain things, and we'll go through a backwards and forwards process in terms of making the clarifications and changes that they're willing to approve. They provide that assessment directly to the Secretary of State. And then finally, I, as anybody else who's applying for a monkey license, will have to put that application through to the Animal and Science Committee, which is an independent national committee that again will advise the Secretary of State independently. This whole process takes about a year to complete. So it's a very rigorous and in-depth and detailed application process that you have to go through in order to be able to work with experimental animals, and especially with monkeys in the UK. One of the conditions of any project license, and in particular for the monkeys, is to be able to use the most refined and proven methods in your research. So my own lab has an interest in doing this, and we've focused on the training procedures that we use, and Stuart's going to talk to you a little bit about those in a minute. But we've also recently developed a protective head cap refinement and wound management control as well. And Brooke will talk to you about that in just a second. So now I'm going to go on to sharing my screen so we can show you some of the research that we do and some of the videos. I'm just going to bring it all up now. So our latest refinements have been detailed in a, sorry, I'm just going to get that right, in a training paper that came out in Journal of Neuroscience Methods last year. And they explain how we effectively train monkeys to be able to enter primate chairs and transport boxes to be able to work in the neuroscience experiments. I'll pass you over to Stuart now.
Okay. So positive reinforcement is when you encourage or establish a pattern of behaviour by offering a reward where the behaviour is shown. Like when the monkey enters the transport box, which you're going to see in the videos. We use voluntary participation, allowing the monkeys to succeed and receive a reward.
Sorry, I'm just going to reduce this down because it's not adding anything.
So here you'll see the monkey he'll come into the transport box. You'll see some collars that the monkeys are wearing. They're a soft neck dog collar, and it has a monitoring chip in the collar, just like a Fitbit to monitor its activity. The transport box needs to be this design for health and safety. And you'll see that the monkey came into the transport box and took the date on top of the transport box. It also needs to have the bars there so that it can reach out and touch the screen and also take rewards from us. I get really attached to the monkeys and enjoy working with all the different personalities. I've trained approximately 80 rhesus macaques using positive reinforcement techniques and a variety of neuroscience studies, and for many lead scientists here at Oxford. Here is another type of transport device box we use. I travel to many countries to present these methods and run workshops for the trainers and the neuroscientists. It is important that the trainer and monkey succeed in the training goal so that both can succeed and know they have accomplished the daily goal. In this next video, we're going to be neck plate. Here the monkey is going to be neck plated so they keep their head above the neck plates used in neuroscience studies. I feel very proud of training and teaching that I perform to help others work with the monkeys and training them for neuroscience research. As a training people to train monkeys is very important. By breaking down the training into small steps, the trainer and the monkeys succeed, and I feel very proud of the monkey when it succeeds. When the monkeys are not in training or experiments, enrichment is a vital component to their husbandry. Here is a group of monkeys having a bath, which is incorporated into when the enclosure is wet cleaned. You'll see they're in a group, and you'll also see one very soon going for a swim. So keep your eyes out. Here we go. At the bottom of the tub is some forage material. It could be dried vegetables, peanuts, or dates. Thank you.
Thanks, Stuart. So we've got several different techniques that we use with the primates, and when we're using cognitive testing to be able to understand how their learning, memory, and decision making is going on. We've got physiological parameters that we use in terms of eye tracking, heart rate changes. We look at neuroimaging, which Juan Carlos will tell you a little bit about in a minute. And we look at neurophysiology, which Brooke will tell you about later on. We also do perturbations in the brain, temporary drug inactivations, or lesions, or microstimulation to targeted brain structures. Typically, we're focusing on the medial dorsal thalamus or interconnected cortical regions. And as Kathy said, we also have stroke patients that we compare things with. We look at human studies, and we look at people who are receiving deep brain stimulation. One of the things that we need to do with the monkeys is put on cranial implants. These are required in order to be able to make sure that their head is held still. For example, we use a head post, and this is a CAD drawing of a monkey with a head post. And this ensures that there's the safety of the monkey and also the humans during the procedures. And I'm going to hand it over to Brooke now to be able to talk to you a little bit about the refinement that we've developed in the lab.
Thanks, Anna. So an important part of what we do is to consistently try and improve the way we do things. So the idea for this head cap, you can see here, came from the work of some colleagues after we found that some of our monkeys picked at their implant margins. So picking can slow down healing and increase the amount of medicines that the monkey may need. These caps, of course, we want to try and facilitate them to heal as quickly as possible. So these caps are cheap and easy to make. And they're made from a special plastic that's used in radiotherapy that can be heated and molded to any shape and used with different sorts of head posts. And this refinement was actually awarded a best refinement in 2019 across all species from the Institute of Animal Technology and the Association of the British Pharmaceutical Industry. And it's just under review at the moment for publication to try and export it to other laboratories. Okay, so now we're going to play a short video or animation video from the Federation of European Neuroscience Societies about the use of animals in neuroscience research.
Our brain weighs just 1.5 kilograms, and yet it does everything. Sensing, thinking, moving, feeling, remembering, and much more. The brain is like 100 billion tiny computers, brain cells, or neurons, each connected to thousands of others. These connections would stretch 4 times around the Earth. Your brain works at lightning speed. It takes 1/10 of a second to fully analyze a visual image. The brain needs energy to function properly, around 20% of what you eat. We need to study the brain to understand how it works. Brain imaging is useful, but scientists also need other methods. Brain cells can be studied in a dish, but this doesn't tell us how the brain affects behavior, and these cells cannot form the complex connections of a living brain. Computer models can also be used, but they are no substitute for the real thing. Studying animal brains is hugely helpful for understanding our own brains. Lots of different animals can be used, from fruit flies to mice, rats, and monkeys. Each animal model has its own advantages, and most of what we know about the brain comes from animal studies. It's important that we continue to improve our understanding of the brain so that we can help treat brain diseases, answer questions on everyday functions such as recognition and memory, and learn about other things the brain affects. For example, why we dream and how sleep aids memory, how the brain controls how hungry you get and what you eat, why you get depressed, and how drugs affect brain chemistry and cause addiction. Understanding the brain can even help paralyzed patients walk or talk again.
Now we've got JC up talking about the neuroimaging.
Well, hi everyone. So yeah, I think one aspect that I would like you to take from this talk, and we as a group are working on what I'm going to tell you about, is this idea that the brain really is working as a collection of networks that interact with one another depending on the task that the person or the animal is doing. So I think we all are familiar with the idea that the brain has different parts that are specialized for certain mental aspects. And there is, of course, some truth in that. However, these areas on their own would not be able to achieve much. They need to interact, and they need to interact to form specialized networks. And the way I see this is that the brain, in a way, works pretty much like a human society. So if you think about the different specialized areas as people, you would have then people that are specialized in, I don't know, engineering, there's musicians, teachers, and so on. But for society to work, these people need to interact really. So for example, this engineer would help his partner to raise their child, or would collaborate with business people to build bridges together with construction workers. And so these interactions in society happen all the time, and they change and depending on what is needed. So it's the same in the brain. And so, for example, if any of these people in society were to fall ill, then they would all need to form a connection with a doctor. Now, an important aspect of these connections is that new information and new properties can emerge. For example, an engineer on his own, or the doctor on her own, maybe would not be able to come up with this technology, with MRI. But together, by collaborating and by communicating with one another, new properties emerge. And the same happens in the brain. So we are going to study the brain as a collection of networks. And a way to do this is to, we can manipulate the activity of one area, of the area that we choose. Say, for example, we give a boost to one area, in this example, to the engineer. We can then study what happens to the networks and to the other areas that are connected to this area, and also what happens to society in general. Will more buildings be built, for example? So by giving a boost to the engineer, we might see that also there's a boost to his partner, to the business people. But we might get surprises. For example, the construction workers might suddenly become lazy. I don't know. We get surprises like this by studying the brain. So to carry on with this analogy, well, to ground it with what we actually do in the brain, what we are doing is training a couple of monkeys to feel comfortable working, doing different tasks inside an MRI scanner that allows us to see the activity of the whole brain. And then what we do is that we manipulate the activity of the areas that we want to study, and then we can see the impact that this has in other brain areas. Say, for example, that we give a boost to this particular area. We study the thalamus. So this is located pretty much in the center of the brain. And we can see then what happens to other areas that are interconnected, or other areas might actually turn out not to be connected. So this is just a video. Yeah, so in this video, you can see the chair in which the monkey goes into the scanner. He has been trained to feel really comfortable while being scanned. And then we have all these set of apparatuses to measure different signals, behavioral, health signals from the monkeys. And then we can see the brain activity, or just the structure of the brain. So the task that this monkey is doing is, I'll go back actually. So basically he's fixating his gaze for about 5 seconds in the center of the screen. And that demands that poses a demand on the brain. The brain needs to put attention to control the muscles of the eyes. And then we can see what's happening in the brain while the monkey is doing that. So these are results from an experiment by a postdoc that was previously here in Anna's group. She stimulated an area in the center of the brain, labeled here as MD, a nucleus in the thalamus. And then you can see that other areas around the brain also increase their activity. So this reveals which areas are interconnected with the area that we're interested in. And so in this way, if we then link this information with the tasks that the monkeys are doing, we can discover which brain areas are interconnected during specific mental tasks and which role each area has. Importantly then, we can then study the activity of single neurons of these areas that we have revealed as important, and that is what Brooke is going to tell you about. So, well, on to Brooke. Or, yeah.
Just me for a second.
Sorry.
Thanks, JC.
Yeah.
One of the tasks that we'll tell you about before we hear from Brooke with the neurophysiology is the object in place learning task. And there's a monkey version of this task, and there's also a human version of this task. And here you can see a monkey sitting at the screen, and he's got the colorful images projected across all of the screen. It's a touch screen. And he reaches out and he responds to typographical characters here and here in order to be able to get a reward. And we're assessing his learning during the task. And here's some more examples in the monkey version here on the left-hand side. This is an object that he has to learn the location within the touchscreen, and also in this background image, it will give him a banana pellet if he touches it and gets it correct. But this one here, if he touches it, he won't get any reward. And then he moves on to other examples within the same session, and he sees 20 of these within a session, and we repeat them 8 times within the session. So we get a measure of how well he is learning, how quickly he is learning, by measuring the number of errors he makes during those repetitions of the trials. And there's also a human version of the task, and instead of using typographical characters, we use fractal images, and here's some examples of these. We get the people, the participants, healthy controls, and also people with thalamic strokes, and the medial dorsal thalamus and the anterior thalamus and in other areas of the thalamus to be able to have as controls, to be able to assess their learning again in a similar setup. But of course, the humans don't get the banana pellets as their reward. And then this is some of the latest research that's come from my lab just in the last week. It's been published in the Journal of Neuroscience. What we did is we had a group of experimental monkeys, and we trained them up in this task. Before we had introduced them to the computers or any training in the task, we did a scan in the MRI to be able to assess how their brain looked before at baseline, we call that. And then after they'd learned the task over about a year to be able to learn the task and get to a really good performance where they were making very little errors, and they were seeing new objects every day, 20 of those each day, and repeated 8 times within the session. Then we scanned them again, and we assessed their performance compared with a group of controls. So we had the experimental group after learning and before learning, and then a group of controls who had never experienced the touchscreen task, never had done any learning and performance, but were just getting rewarded. And we did a comparison of how their brains looked as well. And what you can see here, this is a matrix of the connectivity of certain areas. We had a priori hypotheses about which areas of the brain we would look at. And we can see here the hot colors show increased activation, the type of thing that Juan Carlos was talking about just before, areas that are increased in terms of as a consequence of them having experienced this learning in the task, and the orbital frontal cortex interconnected with other areas in the orbital frontal cortex. And in neuroscience, we know that this area is associated with value and representing the value of rewards. There's also other areas in the medial temporal lobes that are interconnected to the retrospinal cortex, another area that's particularly disrupted. And all of these areas are also disrupted in humans who have learning and memory problems. But the thing that we found particularly interested, we looked at the dorsal medial thalamic region as well, and we could see that there was interconnectivity within that region with entorhinal cortex and perirhinal cortex. So what do these results mean then? What they show is that we have some understanding in terms of mapping this out in humans, but also in monkeys, in a longitudinal study where we can compare what their brain looked like before they had experienced any learning with what it looked like after they had lots of experience of learning in the task. We can understand then how those neural networks are changing the connectivity after learning. And this highlights the extent of the brain plasticity in these regions. The other area that I didn't talk about, these cooler colors, shows a deactivation, reduced connectivity in these regions. And this was surprising for us. We weren't expecting this result at all, but it is surprising. Some areas of the brain are increasing in their connectivity, and other areas of the brain are decreasing in their connectivity as a consequence of learning, because the brain is a finite structure. And like I said, similar to human studies, there were similarities in the frontotemporal connections between while they were learning together. But the human studies do a cross-sectional analysis, whereas the monkeys were able to do the same animal learning across time. And then, like I said, for the first time, we were able to show the subcortical region, the medial dorsal thalamic nuclei that interact with these cortical areas in order to be able to support optimal rapid learning in the task. So then now that we know that these areas are particularly involved and we've done perturbations in the past, then we're also particularly interested in what types of messages the cells are sending. So we've looked at the whole brain, we've looked at the neural networks. Now we're going to focus on what the cells individually are doing, and we'll get Brooke to be able to talk about that. Thanks, Brooke.
Oh, no worries. Thanks.
Okay, so you've seen that one way we can measure activity in the brain is to use fMRI. So another method we might want to use is electrophysiology, which is essentially just recording electrical signals in the brain using small metal contacts. So electrophysiology complements fMRI nicely, as fMRI is great for showing us where something is happening in the brain, but it's less accurate at telling us exactly when it happened. So electrophysiology allows us to see what individual neurons are doing during learning of a task or remembering in a task, and how that might change over time. And it also lets us do it at sub-millisecond resolution, so we can get really precise timing of what's occurring. So neurons use electrical signals to communicate. These are called action potentials. And we can use special electrodes like this one here. And then you can see it is its actual size there next to a 1p piece. Along with special recording equipment, and we can capture these signals while the monkey is running in the task. So as an analogy, it's kind of the same idea as placing one or multiple microphones in a crowd and then trying to record what one or more people are talking about. Okay, so the recordings we make look something like the blue and black traces up here. So the little vertical lines here coming down from the black trace and both sides of the blue trace are neurons firing, and then the thick horizontal line is just background noise. So the waveforms below here, these ones down here, what some of the vertical lines look like when we zoom right in. So we might record multiple neurons on the same electrode, and we use different factors such as the shape of the waveform to tell one neuron from another. So just like in our crowd example, it would be like using the characteristics of a person's voice to tell how many people are involved in a recorded conversation and who said what. So now if we put it all together, we can look at what individual neurons do at different points of learning during our task. So the neurons I'm going to show you are recorded from the orbital frontal cortex, the area that Anna was talking about just a minute ago. And the idea is to collect a sample of these neurons from our region of interest to understand what processes they're involved in. So this data was only collected from correct trials. And if we look at the first plot, the easiest one to understand is probably the second one here. So we can see for this particular neuron that it starts to ramp up its activity around the time of reward. So it gets faster as it starts spiking faster as the reward time is coming up. And then in this example, we can see that this particular neuron recorded from the same region. So these are two neurons doing different things within the same region, is ramping up towards the time the monkey goes to touch the screen and also when the reward is given. So now I'm going to pass you back to Anna, and she's going to have some final comments on why we do what we do.
So one of the exciting things about Brooke's analysis there is showing that over time, in terms of the repetitions of these trials, it looks to be that the monkeys are learning and the neurons are also ramping up in terms of their responses to the learning. Of course, we need to just go further through the data and make sure that that is actually the case, but it certainly looks like it is in this particular example. Watch this space. Well, actually, we'll publish it. So why do we want to learn these things? Why do we want to know how we learn? So we're investigating how these brain changes occur during learning. And then this helps us to identify the neural networks and understand the contributions of specific brain areas. Everybody's heard about the hippocampus and it being involved in memory processing and our ability to be able to learn new information. And that is certainly true. But it's more than just the hippocampus. It's about neural networks that are interconnected to the hippocampus, cortical areas, other subcortical regions, including the thalamus, that are important. So we need to be able to see a bigger picture of all of this information. When we do these causal manipulations, then they also allow us to get an understanding of how this neural network changes over time and adapts, like what we were talking about before about brain plasticity. But some things go up and some things go down because the brain is a finite space. So we need to be able to understand how things are changing. And we do that at the whole brain level with the neuroimaging, but also at the single cell level with the neurophysiology. And obviously then when we've identified these relevant neural networks and structures, like they have in Parkinson's disease, then we can target effective treatments to be able to look at ways to be able to mitigate this the many of the brain diseases that are affecting learning and memory in humans. And that is obviously the ultimate goal, is to be able to support the humans in order to be able to understand and make interventions by using animals. All of the human medical research typically has an animal model in the basis of its basic understanding and fundamental knowledge about what's going on in the disease. So thank you again for your time and listening to our presentation. I hope you've enjoyed the videos and our overview of some of the work that we do related to learning. We also look at memory and we look at decision making in our lab. We're a busy group of people, and we appreciate your time and coming along today to join us. I need to acknowledge obviously the Wellcome Trust for all of their support and funding the research at the moment. The MRC is also involved with a PhD student that I have, and in previous funding applications, funding grants that I've had. And obviously it's a huge privilege to work with the monkeys. They're really special. We enjoy it every day. As you heard from Stuart, all of us enjoy working with the monkeys, and they make the difference. Thank you very much.
Thank you, Anna, and your team for that insight into why and how you're using monkeys in your research. We do have a few questions that have already started to come in. Do fill in your questions in the Q&A section and not the chat box. And as they come in, I will relay them to Anna and her colleagues. Perhaps I can start off with a question, actually. In the wild, obviously monkeys are really sociable and live in quite large and complex social groups. When you've got them living in captivity, are they socially isolated like we are at the moment, or do you have them living in groups?
Yeah, thanks, Kathy. That's a good question and a good analogy to the current lockdown. We have them living in groups in the UK. That's how it's set up. The breeding facilities are in the UK as well, and they're transported to Oxford, and we keep them in their groups. They will typically come as a group of males or as a group of females. So we keep them as the same sex groupings. And then we'll have them either as a pair or in groups of up to 7 or 8 animals. But again, it depends on the experiments and the hypothesis they're generating. But yeah, we don't have singly housed animals unless there is a specific reason why that may necessarily be the case. For example, after a procedure, then they may be singly housed for a period of time. But it's always in consultation with the vet and the animal welfare officer to make sure that everything's fine with them. I'm working on that. So also just to remind you, Kathy, in terms of doing the poll. So while everybody's getting their questions together, we're very happy to answer questions, but we would also like to ask you some questions too. There's 3 questions this time to be able to ask. So please stick around, but also just the same 2 questions from the start to see how well informed do you feel about the animal research in the UK. You can obviously answer the same, but you may want to answer differently. How do you feel about animal research? Similarly, again, you can obviously answer the same, but you may want to answer differently. And then finally, did the presentation help to change your perception of animal research? And again, we won't share that with the overall public, and it's all anonymous. It's not linked to you in any way, but we'd really appreciate your feedback in terms of the questions there.
Great, well, I'll leave that poll running for a few more minutes just to let everyone have a chance, as there are now 3 questions to ponder over rather than just the 2. But I'll turn to some of the questions that we've received. So when you were looking at how monkeys learn and how humans learn, we've had a question: why were the monkeys using letters but the humans were using fractal images?
Yes, good question. So obviously monkeys, we're needing objects on the screen, and the computer task was generated back in the 1990s by the PI that I came to work with, Professor David Gaffin, who's now since retired. Back in the day when computers were just new, and so he was able to use typographical characters, alphanumeric characters, and images are randomly generated. So there's lots of typographical characters and alphanumeric characters to be able to randomly generate as many images as possible so that the monkeys never are seeing the same thing again. Of course, they could see the objects again, which are these typographical characters, but they don't form language. They have their own language, but they can't form language like we do. So when we were developing the task, when the touchscreen computers became available for humans to be able to use, we run the task for the humans on an iPad. Then we were able to use fractals instead, because obviously if it's typographical characters, it's much easier for a human to just form an analogy and go, it was F, and then it was K, and then it was X, in terms of their ability to be able to form these sequences and help them with their learning. Whereas the fractals make it much harder, like it would do for the monkeys, in order to be able to have the two being relatively comparative.
Great, thank you for that, Anna. Our next question is about, you know, we all learn in different ways. So how much variability did you see between different monkeys, and how would you account for that in your analysis and conclusions?
Yeah, that's a great question. So yes, the monkeys do learn in different ways as well. Stuart can probably help you a little bit in terms of getting some insight into how they learn at the start. Just to relay something that is variable about one of the animals is that you would normally think that monkeys like eating bananas, but when I was first in the lab, I turned up from New Zealand in 2004. There was one monkey that didn't actually like eating bananas, so it was very, very frustrating to be able to try and work out what she would like instead, but it turned out she loved grapes, so we were able to give her that instead. But Stuart can tell you a little bit about how we deal with that variability in the initial training, and then I can talk to you about how we do that with the data.
So we like to make sure that the monkey succeeds in its training each day. So the computer tasks that the monkeys are doing is broken down in different levels. So they will do level 1, 2, 3, 4, 5 at the speed that they can work at.
And then, as Stuart indicates, once they get to the isotope in terms of their performance, how they touch the screen and things like that, then we're able to manipulate their learning and the task. Similarly, again, we introduce that in baby steps until they're fully cognizant in the task. And that can take, like I indicated, that can take up to a year for them to be able to do that. So we need a little bit of time to be able to give the monkeys the time to be able to perform. Of course, we have some monkeys who are superstars and have got the tasks sorted within a couple of months, and others, like I said, that it's taking up to about a year for them to be able to learn. And then how we compare that in terms of the data and incorporating in the variability. We tend to convert things to percent scores if we're looking at errors, for example, so we can then make them standardized, or do log scales. For the neuroimaging, we're obviously keen to be able to incorporate what's going on with the different connectivity in the brain, but typically all of those things are done in the smoothing of the data, just like they would do in any study with a human. But obviously we have to be careful with the smoothing. So there's lots of different ways that we make sure that we're measuring apples with apples rather than something else.
That's great, thank you for that. It looks like most people have now voted. I'll leave that vote open just for a few more seconds. You were talking earlier about some of the monkeys, or one particular monkey not liking banana, for example. Have you ever tried other types of reward for the monkeys apart from food?
So currently in the electrophysiology studies and in the MRI studies, we use a smoothie reward. And JC could maybe come and tell us the varieties that he uses for his MRIs.
Yeah, well, to answer that question quickly, yeah, I prepare a smoothie. Sometimes I think that I pamper my monkey more than myself because, yeah, it's a special recipe with some Rice Krispies, with apple, with some ribena, which is this black currant squash, and water. That's what these monkeys seem to really enjoy. Yeah.
Thank you. We've had another question asking about, you know, how often and how many times you can run fMRI scans on an individual monkey. Is there a limit?
Yeah, Well, the details of the license actually Anna will be able to tell you more about, but that's definitely one of the advantages of using monkeys, and that's why, yeah, we use the monkeys instead of humans, and is that they can have many sessions, and actually during a session do a lot of trials. So one of the monkeys I'm working with at the moment, sometimes in less than an hour can do maybe 160 correct trials, and he's happy to go into the scanner every day, well, 5 days a week. So that obviously is a huge advantage because it gives us a lot of data that we can then study.
That's great. And how does a monkey brain compare to a human brain?
Well, obviously we are using the monkeys because their brains are very similar to a human brain insofar as their connections and their anatomy, but at the same time there's many areas that are underdeveloped, let's say, compared to a human. So it's a little bit of a trade-off. We need a species to study that is as close as possible to us, but at the same time, we need to be always aware that it's not exactly the same as a human brain.
Can I just add something there, JC? So also, importantly, we use the rhesus macaques. They're an old world monkey because they have a very well developed prefrontal cortex that isn't exactly the same, but is very similar to the frontal cortex of a human. And the areas of the brain that I'm interested in the medial dorsal thalamus and the anterior thalamus, are interacting with the prefrontal cortex, and they do that in a very similar way across the species. The other types of neuroscience that you can work with the monkeys is visual processing, because like the monkeys, humans, obviously we're the same in terms of our visual processing. We both see in color, we have eyes at the front of our head, and we have binocular vision. And the other thing that we do is reaching out and grasping things. And similarly, again, the motor circuitry in the brain of the monkey and in the human is very, very similar. And again, with the spinal cord going down the back and your ability to be able to work hand-eye coordination and foraging in tasks and working hand-eye coordination, especially on your computer, all those types of things is very similar across the species. So those are the areas that we're particularly interested in, and they're very, very similar. Obviously, there's not as many neurons in the monkey brain as there is in the human brain. So it's a size component there as well.
Right, well, that brings me on to another question that we've had. You mentioned that certain neural connectivity is reduced as the monkeys learn. What are the implications of that?
Yeah, so what it suggests is that those, as the interconnections that we are seeing in the orbital frontal cortex suggest that the monkeys have learnt the task, they've learnt the value of the rewards of the task, and they're primed and learning about things that are important to them. And then other areas that are showing a reduction in connectivity as a consequence would suggest that those areas aren't necessarily, it's not to say that they're not involved, but they're just not, this strength of connections isn't as increased as a consequence of them learning in the task. One of the first people to show this was Eleanor Maguire in the famous taxi driver study that she did of London taxi drivers who'd learnt to do the knowledge, the black cab drivers. So while she was able to show that one area of their hippocampus improved as a consequence of them passing this knowledge test after 2 years of driving around in London, another area of the hippocampus shrunk as a consequence. So it's like anything, there's always a push and a pull, and we've only just been able to start to understand those kinds of things in neuroscience in the last 20 years as a consequence. And it also works in a similar way in terms of excitatory connectivity and inhibitory connectivity. So the consequences are that you're always trying to create some kind of homeostasis in the brain. And we've only ever looked at the things that increase because we think that bigger is better, but sometimes the things that decrease can also be just as helpful. And presumably they're doing that in a way that then maintains the optimalness of the circuitry, so that we're not then going on to develop seizures or something as a consequence of everything just firing and rewiring to the max, because that isn't actually the most optimal, the way that the brain works. It's always about some kind of a homeostasis and finding the balance. Great.
So there really is only so much you can hold in your brain.
Yes, exactly. Yeah. And many things that we don't know, we can only attend to so much. We can only learn so much. We'll often think on, you know, like we're doing now, we're thinking on the spot and we'll think, oh, I wish I'd said that as well. But you can only do so much at any one particular time. You can only make the optimal decision based on the information that you have in the here and now.
Right. So earlier on, you were using the comparison of the brain with society and how if one part of the brain is stimulated, it then affects others. But what would happen if the engineer in your analogy died? Could they be replaced?
Well, so that's what we do as well. We look at manipulations in the brain, causal manipulations. We can do that in the monkey, and also obviously compare it with people who have thalamic strokes or people who have neurodegeneration, Alzheimer's disease, frontotemporal dementia, neurodevelopmental disorders like schizophrenia. So yes, all these things change the brain in different ways. But if somebody was just to say instantly die, or an area of the brain was instantly to die, then that would be occurring as a consequence of a stroke. So they're a very good analogy to the type of work that we're doing in the monkeys in terms of these instantaneous changes. And what we find is that if we're targeting the medial dorsal thalamus, my hotspot of a brain area that I'm particularly interested in, then learning isn't optimized any longer. We can learn, but we do it very slowly, and we can't optimize our learning, and we can't optimize our decisions any longer. So what it seems to be, and that's what we're looking at now with the neurophysiology, is that the frontal cortex needs interactions with the medial dorsal thalamus in order to be able to make the most of the learning environment that we're in. And if we don't have a medial dorsal thalamus, if that's the part of the brain that's died as a consequence, the engineer's died, then we're missing out on the ability to be able to link the things that we're learning about in our environment in the here and now, to be able to optimize that and making our future choices and future decisions.
Can I just quickly say that I think this is also one of the key goals, that by understanding, for example, with what Brooke is doing, the recordings, he's understanding the code that the cells are using to communicate with the prefrontal cortex and pay attention and form memories. So eventually, and this of course is going to take so many years, but the goal eventually is that we will be able to substitute this structure in case, let's say, the engineer dies.
Cool. Thanks, JC.
Thank you, JC. Just looking at a couple of the other questions. So are the monkeys actually able to teach each other things? I mean, for example, do they swap tips on how to get the rewards?
Sometimes we think they do. So from the videos, well, maybe not so much from the video that you've seen today, but certainly the one that you may have had a look at beforehand, where we're showing them in the Welcome Trust video. They work in individual cubicles. And the type of research that I do, there's another group in Oxford who do social neuroscience and look at how interactions between a pair of monkeys is going on. But we're looking at individual performance. So we have each monkey in a separate cubicle, in a separate setup, while it's doing its cognitive testing for the hour or 2 hours that it's involved in the tasks. And obviously only one monkey can go into the MRI scanner at any one time. So it's more that when they go back into the home enclosure, they potentially are somehow talking to each other and working out the best options. But one of the things that we've found in terms of the effective training is that they teach each other. And Stuart can talk to you a little bit about this further.
So when we were having the monkeys come into the transport boxes, you could see when another transport box turned up, the monkey that was going to come in there was getting very excited because he knew that going in there was going to get a reward, because he had seen the other monkey take the reward off the top of the transport box. So they're watching, they're very visual. So even when you're turning up on your daily work with them, when you bring in the transport box or the chair, they get very excited because they know they're going off to the computers to use the touch screens, get the smoothie reward. So they get very excited and they want to be next.
Oh, it's great to hear that they actually want to do these things that you've trained them so well to do. How many monkeys do you actually have there, and what issues arise from perhaps having a limited number of individuals going into those hour-long experiments?
At the moment I have 7 animals in the facility. The other groups in Oxford contribute obviously to that as well. There's at least 4 PIs. I'm one of the PIs, and they all have groups of animals as well. We all have staff. Obviously, we can't do anything by ourselves. Nobody can in science in terms of collaborations. All of these things are important. So you've met some of my postdocs in RA today in terms of the people that are working with the animals. I also work hands-on in the lab most days as well. It was hard over the lockdown. We obviously still had to come in and maintain the monkeys. We have technicians who do all the cleaning of the cages and things like that. So it's a real team effort. We have the vets involved. They come around every day. They do a morning check, just like checking everything's fine. Then we have an animal welfare officer. They also come around every day. So it's a real team approach in terms of working with the animals, and lots of communication between everybody to make sure that everything is as optimal as it can be for the animals, because we need them to be stress-free. We need their welfare to be as important as we possibly can in order to be able to ensure that we're doing good science. So, and the kinds of things that we're looking at, so the manipulations we do within the animal. So we are able to compare what things are looking like before, like you heard in the scanner, before they ever start learning in the task, and then how their brain looks after learning in the task. And that's the uniqueness about the monkeys. So we have that within animal comparison. You can't do that in humans. So that's why you need so many more people. You need a cross-sectional analysis of those. And as you've already heard, there's lots of variability in the animals. So that already adds to that randomization without needing to be random. Of course, we don't pick the monkeys ourselves. They are randomly sent from the breeding center for us to be able to work with. And then in the neurophysiology, typically we will have 2 animals in each experiment, and we're actually looking at the neurons, the units, the spikes that are talking, and we do the analysis on the number of spikes. And Brooke can tell you a little bit more, but I can also obviously tell you we will typically have a session of recording where we're collecting maybe between 20 spikes or 30 spikes, depending on the number of probes that we've got in and the number of contacts on each of those probes, anything up to about 120 spikes. And then we combine that, those spikes over sessions of recordings. So we're getting up to 1,000 spikes that we're then doing the analysis on, and we're comparing within the task when the neurons are firing at certain particular times to be able to understand what's going on.
Right, so you just mentioned about what a large human team you've got working with the animals and how lockdown has caused possibly issues with that. How do you think the pandemic will affect monkey neuroscience research in the UK?
Well, fortunately, the funders have been supportive. The Wellcome Trust is looking at extensions for our funding at the moment. So hopefully things will go ahead to keep going. There is a concern, obviously, amongst younger PIs who are just trying to establish their labs, but also PhD students, early career researchers, postdocs, in terms of the opportunities for jobs, to be able to change labs, to keep working in the future, what's going on. It's a concern for all of us, obviously. We're sort of 3/4 of the way through the funding that we have. So it's like anything, obviously everything is always a worry. But the facility we have is great. The monkeys have been well looked after. We're involved in well looking after them, and we've got different bubbles of teams in case any bubble was to go down with COVID, because obviously monkeys are susceptible to the virus as well. So we've had to work really closely with the vets, and they've got information from all around the world to be able to ensure that that everything is as optimal as it can be in order to be able to support it. So we're hoping, and that's all we can do in a sense, keep our heads down, keep working, keep getting the results, doing a great job and supporting the animals and the work, and things hopefully will continue. So
I actually didn't realise that the monkeys were susceptible to the virus as well. Is that because they're so closely related to us?
Yes, they're one of the species that is used to be able to test the antibodies, the vaccines, all of that. Ferrets are also used, and mice models too, but the mice have to be genetically modified. So ferrets and monkeys are actually susceptible to coronavirus as well. So they're being used here in the UK and all around the world in order to be able to help push these vaccines to be able to be sorted for everybody. So all of this work, the infrastructure of all of this work, where neuroscience is a small part of that in terms of vaccines, immunotherapies, the flu virus every year, all those types of things, polio vaccines, everything is researched in animal model. All medical research is researched in any kind of animal model. It's the particular animal model that is each different type of animal model is used for a particular medication because it's the most suitable for that particular example. For example, mice and rats don't get coronavirus, so there's no point checking the virus in them in terms of the vaccine. Then monkeys are needed for that.
Right, that's really, really interesting.
Sorry. That was an confusing answer. I'm sorry about that.
We're a couple of minutes over. Are you happy to stay on and answer just 2 or 3 more questions?
Yeah, sure. Yeah, I'm more than happy. Yeah, thanks.
Right. So we've got one that's sort of specific to the cap that you're using and the head post. Somebody has asked, have you ever tried using a thermoplastic face mask instead of using the head post?
Yes. So those are colleagues up in Newcastle who have developed that, and also colleagues in Lyon in France who've also been involved in developing those technologies. And we were aware of them when we designed the head cap. Do you want to add anything? Or?
Yeah, it's definitely in development. We just haven't quite got to the stage of trying it out yet. But it is a refinement that we want to try in the future to see if it will work, because it would be a useful thing to be able to have in our little box of tricks to be able to.
I guess far less invasive.
Yeah.
Yes, exactly. But even in Lyon and in Newcastle, where they were developing it, not all the monkeys were able to cope with using the thermoplastic mask. So, and we do have to use all of the animals in the UK. It's not possible to just have them sitting around and not being in use. So yeah, and the longer term, it's something that we want to use. But in terms of the initial development, we need to be able to get on and make use of the monkeys in the most refined way. And for us, and in order to be able to use all of the monkeys for now, the most refined way was to be able to implant the head post and then develop this cap to be able to protect them.
But I guess over time you'll be trying different methods and trying to find the one that is the most humane to use with the monkeys.
Yes, exactly. All of these things are always a work in process over time. So, yeah.
Great. Just 2 more questions. We've had a question, how many years will a monkey be part of your studies? And then when they retire, what happens to them? Do they stay at the university? Is there a retirement home for monkeys? Yeah,
so great question again. So in the UK, a project license will go for 5 years. And so typically, although not always, a monkey will be around for the length of that project license. But sometimes if we can apply to the Secretary of State and they're willing to move them on to the next project license, assuming that you're going to get one and you need funding obviously to be able to keep the animals going, then they can stay around for a lot longer. But we have a regulation in the UK that as soon as they've received their first experimental procedure, then they must be gone within 10 years of having experienced that first procedure. So they can never be around for more than 10 years after their first experimental procedure, essentially. And in terms of the neuroscience research that I do, we actually perfuse the animals at the end. We put them down and we actually extract their brain, because what we do is a lot of ex vivo studies on the neuroconnectivity afterwards to be able to look at the whole brain and the connections between things as a consequence of what we've been doing while the monkey is alive. So we actually make use of the animal in vivo, but also ex vivo, in order to be able to understand how their brain has changed and look at different things, different immunohistochemistry and different staining techniques to be able to understand what's going on. So some other monkeys are retired, typically not in the UK, but certainly in Europe. Other monkeys are retired to different areas. different facilities, zoos, for example, will sometimes take them as well. So it just depends. But obviously, if they had an implant, then that is removed. And again, you would need authorization from the institute to be able to ensure that that was the most ethically viable option for that particular animal. It's done on a case-by-case basis. But in the UK, typically they aren't retired. They are used afterwards as ex vivo models.
Right. So
just to say as well that the other parts of the monkey are also used by many other groups. It's not just the brain. Many other organs in the body are used by many other groups in Oxford, but also all around the country, and we sometimes ship them into Europe as well. So they're a very valuable resource in terms of the privilege of being able to get access to this tissue afterwards is very valuable for so many groups. So we're more than happy to share it in terms of making it as viable as possible.
Yeah, so you don't want anything to go to waste.
Absolutely not, no.
And so somebody has asked whether the head post would be removed after the experiments, or if you're going to sort of put the animal down at the end, is it just left in place?
Oh no, absolutely, it would have to be returned, and returned to not having a head post on its head, and that is surgically removed, and then the skin is resutured over, and again if that was to happen here in the UK, we would need permission to do that. Of course, we don't have that at the moment because it's not something that we do, but we know that they do that in France. Colleagues in France do that, and there's no adverse effect of that as a consequence.
Great. So the last question we have is actually a comment, and it's something that I would like to relay to you, that your monkeys are clearly very lucky to be living in such an enriched environment and to be such willing workers, but also to have such a team who clearly care very much about them. So it really just leaves me to say thank you so much for sharing your knowledge, your behind the scenes tour of the lab.